Hi, good morning. We are pleased to have Alex Matov from Cornell University, which is actually in New York City, College of Medicine. He got his PhD from the Scripps Research Institute in San Diego. So he's going to talk about his recent work in fluorescency microscopy analysis and tracking.
Thank you for the introduction, Dr. Shah, and for the opportunity for me to present your work and discuss it with you guys. So I'm a research associate at Cornell Medical in New York, and I did my PhD I did my undergrad in engineering, electrical engineering, and my PhD in computational cell biology, where I work a lot on computer vision algorithms for segmentation and tracking. So my talk will have two parts. First, I'll talk about the tracking fluorescently labeled proteins in living cells. And the second part is analysis of circulating tumor cells in blood samples. So first, I'll start with the tracking part. This is a time-lapse microscopy movie of a dividing cell. So what we see here on this movie are the chromosomes. And during the division, they're segregated into two new cells. So we see here the chromosomes. This is the DNA in not condensed form at the end. So that's a looped movie. The DNA gets condensed into chromosomes. In the human, they will have 23 chromosomes. They're all duplicated during division. We have 23 sister chromatids aligned in the metaphase place, and they are then divided into two new cells. So this is one of the most fundamental processes in living organisms, the cell division. And a cartoon model of the mechanism and the structure that does the division is the mitotic spindle. So the mitotic spindle is built by, it has two poles on both sides, and is built by microtubules. The microtubules are these fibers. These are polymers. The cytoskeletal proteins. They consist of alpha and beta heterodimers, which are connected, linked into elongated cylindric polymers. Actually, in this cartoon, the microtubules are probably bundled in dozens of them together to form these structures that elongate and connect to the chromosomes. Here, for simplification, we have like a simple simple cartoon with one duplicated chromosome. Actually, in real life, there are 23 of them. And so what happens is that during division, the chromosomes are segregated with this structure of this mitotic spindle. And normally, traditionally, in cell biology, people have analyzed microtubules and their behavior by such movies like this one, when we have continuous labeling of the whole microtubule lattice. So what you can observe by this movie immediately is that it's very hard to see, for example, in the middle when we have high density of polymer. And so the type of movies I have been working on are movies obtained by fluorescent speckle microscopy technique. So this technique was developed in the late 90s in the lab of Ted Salmon in North Carolina. And what it relies on is on a very low labeling ratio of the tubulin dimers. So what we have here is, again, the traditional movies that I just showed you, where we have about 20% or more of the dimers of the polymer labeled. And then in the fluorescent speckle microscopy, we have about 1%. So what this allows us to have is a fiduciary marker of polymer translocation or of polymerization. So you see the difference between this and these images. And so if we have a movie obtained this way, what we have as a result is we see the same structure of the mitotic spindle during division, and we have a movie like this one. So what we have on this movie is, again, on two sides, they're not labeled. We have the poles. The chromosomes are in the middle. Actually, they're here when we don't have the signal. And these are the microtubules. And these speckles are the signal that we want to detect and track and analyze. So what are those speckles? The speckles are sub-resolution clusters of fluorophores. So what we have in these images, we have an imaging of, with a high resolution microscopy, where we have numerical aperture of 1.45, which means that the limit of detection of two adjacent spots is about 250 nanometers. And so within this distance, when we have between 4 to 7 labeled subunits, we can see above the noise floor a signal. So again, these microtubules that we analyze and we track, they are made of these alpha beta heterodimer subunits. So they have polymer subunits that are linked in a longitudinal way. There are 13 of them that form the cylindric microtubule. It's hollow in the middle. And then what we have, actually we have bundles of many of those microtubules next to each other. And then again, if we have this cartoon, we have again the two poles, chromosomes are in the middle during cell division, and we have the microtubule bundles. And so what happens here is we have antiparallel flux. And this antiparallel flux is difficult to analyze because we have this motion that is fast in comparison to the distance between the particles. So you see on these antiparallel threads here, these tracks there where the microtubules move, or the speckles move, they're very close to each other, but the displacement from frame to frame is very fast. So what I worked on was algorithms to track such images. So talking about tracking, generally speaking, if we consider this simple scenario when we have three spots in frame 1, and then let's say 3 in frame 2, a normal tracking scenario would maximize the number of links by forming candidate matches. We have a certain cost of linking association. Let's say we have, in terms of the nearest neighbor, we have the simple Euclidean distance. Then we form these links. We have the third frame where we have, let's say, 3 more particles. Nearest neighbor would make such a link. So we can appreciate immediately that in the case of these microtubules that are rigid polymer structures, we cannot have this scenario. So we would like to have a solution that offers such links. But also in the case of these spindles that I just showed, we know that they have this anti-parallel motion. So such a scenario when we have 90 degrees would not be possible. So basically what we need here is to introduce a local and also global models of motion. So what I worked on is development of algorithm based on graph theory when we have the scenario of having flow from source to sink. We have these triplets in frame 1, 2, and 3. All the links have arcs have capacity. Here is 3, and from the triplets to the individual particles is 1, and then all of them have cost of linking. To reduce the combinatorics of the solution, I used the search radius that is based on biology, so we know what is the possible maximal displacement between frames. And so the next step in this was that even if we can obtain let's say max flow mean cost solution of that, so from all possible solutions of these triplets we can find the one with the minimum cost. What I observed is sometimes in these movies, it's the best way to approach is to have a trade-off where we sacrifice a certain flow to get the best possible triplets. So this is illustrated in an example when we have again the simple scenario: frame 1, frame 2, frame 3, frame 1, frame 2, frame 3, frame 1, frame 2, frame 3. And again, in this, the cost of this linking, because of this high angle, and also they're not equidistant, will be very high. But if we give up one level of flow and we have 2 links out of 3, we're going to gain a lot in terms of reduction of cost. So the way I solve that is to solve a bi-objective optimization problem, or we have on one side the reduction from the maximum flow, and on the vertical axis the total cost. So what we have here basically is a Pareto front. That's a convex problem. And we have this maximum cost at no reduction, so the total cost at the maximum flow, sacrificing some flow and then having less total cost and then we have completely. But the idea here is that we have this utopia point at 0, 0, right, which is the perfect solution. We don't sacrifice flow, and we have no cost at linking. This is not possible to happen. So in terms of solution, what I found was the closest point on the curve, which is the point here. And then
so in this one, like, how are you computing the flow? What you said on the x-axis you have flow.
The reduction of the flow. So at the very end of it, here, we have the maximum flow. So in this case, it will be 3. All the particles are linked with particles in the next frame. And then here is the no flow. I mean, it's here, sorry, I made a confusion. This is the horizontal, here is the flow reduction. So we have the flow reduction of 100% is here, so we have no links and no cost. And here no reduction at all, we have the maximum close. So everything here goes in between. If I sacrifice one layer of flow or one link, I'm gonna have a drop in the cost, and it keeps going like that on this curve until I have no links at all.
So I mean, it's basically measuring that if you are able to track or not, right? So you have these different particles, different frames. You are saying if they are linked to the next, particle or not, but it's not looking at whether they are correct or incorrect, or are they also looking at the correct connections?
So we have no ground truth in these links, so we don't know if they're correct or not. But
they have to be connected to something.
Yes.
And if somebody is not connected, so then the flow is reduced.
Correct.
Okay.
So here, for example, from flow 3, we go to flow 2. It could be 1 and 0. And then for every scenario, we sum up the cost. And then, actually, in order to approximate that, what I looked at was the distribution for one solution of all the costs. So that's the maximum mean cost. And then created a cutoff at where the cost started having large numbers. And then enforce this to the flow. So instead of having, let's say, a flow of 1,000, I would have a flow of 700, 800.
And the cost is the Euclidean distance between, like, what's the cost?
The cost is the Mahalanobis distance between the 3 elements, which is the angle between, so this is the triplet, frame 1, frame 2, frame 3. So what we have here in first, this is the mitotic spindle. So we have the 2 poles, chromosomes are here, and they flux up and down. First step is the detection, not talking about this today, but basically it's the statistical helpful significance of a local maximum above background. And then the cost for linking is the angle between the 2 segments, and then the difference between the length. And then what I obtain is this row links from head to tail. And then I cluster them, and I find the main directions of motion. So here all the red vectors go up, and all the yellow vectors go down. And after the clustering into those, so I assign each of them to one of the 2 groups. Then I do anisotropic filtering, because I know the main direction of motion. So I can average every vector by the vectors that are before and after. And by this way I obtain these flow fields, upward and downwards. And then the third element of the flow of the cost function is basically the global. So we have the local and the global motion model, which is basically how much is each of these vectors deviating from the overall motion. And for all of these elements of the cost function, I have certain distributions. And the variance would give me the independent random variables. So then the variance will give me the weight of each of them. And this way, it's self-adaptive.
So the delta S1 minus S2, what is S1 minus?
Oh, these are these distances. So basically, in the case of the spindle, we prefer no acceleration. If we're equidistant, the cost is low. So this way, this is also not limited to 2 directions. It's self-adaptive cost function, and normally it converges within 3-4 iterations, where less than 5% of the links are changed. And so I actually evaluated the performance. Some of the sets were taken from ULab here, and from this paper in PAMI a few years ago, the rotating piece, rotating ball, the flock of fish and birds. And you can see the performance here. I don't have numbers to that, but it was the links were good. So again, we're here just tracking flow. We don't have full trajectories. It's 3 by 3 frames. And then actually the most challenging set, I benchmarked this algorithm. It was taken from a Japanese group that generates these PAV datasets, where we had the distance, the mean displacement from frame to the next was actually larger than the mean nearest neighbor of particles within the frame. So this is the set. We have three frames, and I perturb about 1/3 of the particles. Because basically the key of this algorithm is its ability to fish out from noisy data the overall flow direction. So we have very high level of specificity and not so high level of sensitivity. So you see here, for example, when I'm plotting here, that we have competition. Some of the particles participate in 100 possible triplets. And so what I used this in terms of technical application was to initialize a Kalman filter. So a linear Kalman filter, right? Because it can give you on the fly the instantaneous orientation of the flow. Another application was to study these very fast evolving structures, again, this mitotic spindle. So we have what we see on the movie here is a mitotic spindle again, skewered by a needle. So there is a needle here, and the spindle is just avoiding and leaving this needle. You can see how it's going to start again. So this structure is highly dynamic and changes the direction of motion. So it's very hard to track. Plus there is anti-parallel motion. Another application was to study three-directional overlap flow. So that's the same scenario. The only difference is here that part of the structure was glued to the cover slip during imaging, so we got like a third direction. So I could resolve the 3 flows. And the last example I will show you from these spindles is actually a monopole spindle. So that's a collapsed spindle when we have one single pole only. And I don't know if you can see in the movie, but we have very faint inward flux.
So in this movie, you are assuming the safe locations is known, like the detection is done, or?
I detect the speckles.
And so you have a separate method to detect these?
Yes.
And then you do that?
And then track, yes.
Can you ask a couple questions? First, do you know that the speckles that you're tracking in successive frames are the same exact points in the actual object? Like, are these points the same, or because the way you explained initially, it seemed like there are subsamples of the actual structure?
Yes. So this is basically, it's not defining a unique fiber. And also the problem is that you remember how they're formed, so they get incorporated into the lattice, but they also fall off. So basically what you can have is in one pixel, you need to have like maybe 5 or 6 of those at the same time to pop up to see above noise. But if some of them would leave, you lose the feature, right? So it's very...
So it's possible that the points in one frame are not exactly the same set of points in the next frame?
I don't understand the question. Like, I mean,
I think 100 points in the video. Is it going to be the same 100 points in all the frames?
No. They last for like 3-4 frames. They constantly appear and disappear. The one thing that is in common is that we have this translocation of this polymer. So we have the motion, but the motion, the participants, right, they're appearing and disappearing all the time. So one thing...
I use them for 3 frames.
Right. And initially in your algorithm, you were clustering into 2 flows. How do you get the flow vector? Do optical flow or something?
How do the clustering?
Yeah.
I use expectation maximization to find the number of directions, right? So you can see how many how many overall directions. If you plot the angles, right?
How do you get those direction vectors, the flow vectors?
From the correspondence. First, you
But that's the problem he's solving.
By expectation maximization, you find the directions and the number of classes, let's say 3. And they go 0 degrees and 90 degrees. Let's say 2, just to make it simple, 0 and 100. And then I do k-means to assign.
Right, but the points that you're clustering have the direction information. Right? So you're saying, cluster into 2 directions.
Mm-hmm.
Where do you get the direction?
From the expectation maximization, for example.
Then after the clustering,
Oh, so it's like in the iterative process. So initially you don't have direction, but then you...
No, I make 3 or 4 loops.
Okay.
Right.
So in this one, like detection of these points, is it much harder than tracking, or tracking is harder?
I worked on 2 algorithms for detection. One of them was, for example, difference of Gaussians. So if we have with the narrow Gaussian filtering, we get the high frequency noise of the background out, and then with the Gaussian with wide variance, we have the overall background in the fluorescent out, and after subtraction of the 2, we have a bandpass filter. All of them pop up. So detection is not that hard. But the problem is that the features are not stable, so they appear and disappear. So you cannot keep. You don't know there is no constant number of them. Okay, so now the same speckles and the same kind of analysis in a different protein structure. So actin and microtubules are the 2 main cytoskeletal proteins in all cells. And for example, actin is the protein that is very active in cell protrusion. For example, in wound healing, when we have a cut and the cells are moving to close the wound, the actin is the one that forms networks at the tip of the cells. So this is the cell, this is the nucleus. Microtubules are over here that we showed before, and the actin is the leading edge, and it protrudes, then it adheres to the surface, then there is a traction and contraction, and the cell moves forward. So that's also fundamental problem in cell biology. And there is a very complex actin structure of many proteins involved in this contraction and this motion. And then we can use the same tracking and the same also imaging in terms of speckles to analyze actin networks. So that's again a leading edge of a cell. Instead of microtubules, we have another protein, actin. And I worked on an algorithm that was using neural networks and a nearest neighbor to do this tracking, and you can appreciate that there are errors here. So we have this, I don't know if the computer is overheating, but it's getting a little bit slower. So what we have here is anti-parallel flow. We have motion retrograde up and anterograde down. And here there are yellow arrows down and up, and over here in the middle, they kind of cancel out, which is not the correct tracking. And then I used the same algorithm with the 3 frames to track such behavior, and I found that the networks that basically overlap, they're interlaced, interdigitated. So this is a small insert. I don't know if you can see the anti-parallel flow by eye. And then actually this is something that we started working with Sarfaraz to analyze such contractile anti-parallel actin networks. So in traditional methods, again, think example of the nearest neighbor. When we have this network motion that you saw before that is anti-parallel, if the measurements are incorrect, we have a canceling out. So we have here the speed map. This is high speed, this is low speed. So this is published in a previous paper with another tracker where the convergence zone is not correctly tracked. So this is one objective I wanted to see here with your lab, is to see if we can have different trackers and different approaches to dissect these anti-parallel zones of converging networks. And the last examples from this flow tracking I'll show is in the case of the growth cones. So the growth cones, they're part of the developing axon. We have here scenario, I took actually this from Wikipedia. So this is the axon the growth cones, the green parts are actin filaments, and these are the microtubules again. So this is the axon is a part of neurons, and the traditional view of this, the behavior of the actin in such cells is that at the neck of the axon there is convergence zone. So basically there is no motion. So if you see I don't know if you can see this in the movie, but what I measured, so this is the cartoon of the growth cone. What I measured with this algorithm was that we have anti-parallel flow. One is going inward, one outward, and they have different speed distributions and different speeds, yes. And so this is the end of the tracking part. So I'd like to acknowledge all this work was done in the lab of my PhD advisor, Gaudens Danuser, and most of the movies, so most of the microtubule movies are from the lab of Ted Salman in University of North Carolina, and most of the actin movies from the lab of Claire Waterman. Now, next thing I would like to present is the analysis of circulating tumor cells from blood of metastatic cancer patients. So that's much more translational research and cancer research-oriented project. So we're shifting here gear a little bit. Okay. So circulating tumor cells are basically cells that have escaped an organ and flow in our bloodstreams, rather the bloodstreams of people with metastatic disease. So they have been used a lot in oncology as a source of so what they call liquid biopsy. So normally when people want to study in oncology tumors, they take biopsy, which is a very invasive procedure. It's not always possible. And the tumors, these circulating tumor cells, are an easy way to assess a tumor because they can be obtained just from drawing blood. And so there is only one FDA-approved method of counting and enumerating these cells so far in the States, which is the cell search of Veridex, which is Johnson & Johnson company. And they use a certain molecular marker, or EpCAM, basically something that would mark the tumor cell. And basically what they do is they call it blood sample enrichment. So they draw the blood and then flow through a machine where the machine collects only the tumor cells and let the other cells go through. And then, and here comes the vision problem, we have a positive identifier of tumor cell, we have a DAPI, which is stain for the nuclear of the cell, and we have a CD45 that is a stain for a blood cell, which should mean that the tumor cells are negative for that. And so what I've been working on this, in this context, is instead of having this sample enrichment that is supposed to collect only the tumor cells, to image all of the cells, so to have no enrichment. Because there has been a lot of problem with this machine in terms of purity, and there are many false positives, up to 90%, so it just misses the cells. Once they're gone and we don't collect them, we cannot do any analysis. So the idea was of this project is to develop a computer vision algorithm which can serve as a diagnostic tool. And then what we want to do is to predict response to treatment of microtubule targeting drugs. So the microtubules that I talked to in the first part of the talk, they're also a main target in chemotherapy in most of the solid tumors, like prostate cancer, breast cancer, ovarian cancer, gastric cancer. And so the circulating tumor cells, these cells are imaged by multiplex confocal microscopy. So they draw blood and have stains of fluorescent stains in 3 different channels. DAPI stains for the nucleus, prostate-specific membrane antigen would stain the prostate cells, and the CD45 stains only the leukocytes. And so, additionally, besides for just counting these tumor cells in a volume of blood, what we can do is in high-resolution images to evaluate. So these are fixed cells. These are not living cells. They're not moving. So we have just these one images. This is a cell in microtubules before treatment with microtubule targeting drugs, and this is after treatment. So there is changes in the image. And then In one sample, normally we have, let's say 50 images, 50 positions. What I'm showing here is only one position, and this is the problem I've been working on, is to segment these images and make a selection of which one is a tumor cell, which is not. So in this case, we have the PSMA, so that's the prostate-specific membrane antigen, so this would show the tumor cells. You see that the stain is not perfect, so there are a lot of non-specific staining. This is the DAPI that shows that there is a cell, so it means there is a nucleus, and this should be the cell CD45 negative, meaning that if we have this label here, this is a blood cell, not a tumor cell. And on the lower part of this, I'm showing a segmentation of these images using a wavelet algorithm that I used. So I used the wavelet-based segmentation in order to denoise the images and to obtain only clusters of intensity. So this is an algorithm developed by Jean-Christophe Oliveau-Marin in Pasteur in Paris. And what we have here is from an image with a lot of background. After denoising, we have only the spots with the bright intensity, the clusters. And I've been looking into computing some morphologies and size and eccentricity of these areas in order to be able to guess or to identify the spots representing tumor cells. So one thing I try to do is work into automated unimodal thresholding, so we have distribution of certain morphology, let's say area of centricity, and then automatically select a cutoff. But this has not been working universally of all the images, so there is a lot of heterogeneity. And so this is something I would like to discuss with you here. One algorithm that I identified for such segmentation is developed in Texas. This is the article when they have this automated detection and segmentation of nuclei. It's a multi-step algorithm based on Laplacian of Gaussian filtering. The second part of this project is to image this. So the first part is detection and enumeration. So we have a very clear readout there. When we have a patient that is treated with certain chemotherapy drugs, he has less cells after treatment, so that's a positive response. But another thing is, after we find the coordinates of each individual cells, we can actually re-image them at high resolution. So the first imaging part was at 10x, and this is 63x, so high resolution of imaging of the microtubule network, where we can evaluate changes in this network after treatment with drugs. So, and so basically use the microtubules as a biomarker of treatment efficacy in real time. So these drugs that I've been talking about, the microtubule targeting they have several types. One of them that is used in prostate cancer are the taxanes. So they stabilize microtubules, and they have been used in oncology Taxol has been used for 20 years. Since 93, it's FDA approved. And I think there have been probably a million patients that have been treated, and there is no way to predict if it's going to work or not. So there are different types of resistance, and the main problem is that even if it's a validated target in oncology, there is resistance, and that hampers the success. So it was initially obtained by a tree, but now it's synthesized in the lab. So what we would like to do is assess changes into the microtubule network after treatment. For example, in cells ex vivo, so the patient is not treated, just the blood is drawn, and we can see whether a cell after treatment with the drug is changing or not. So you see here, resistant cell does not change, and let's say a sensitive has these bundles that we can quantify. And so what I use on this context is a platform developed by Robert Murphy in Carnegie Mellon University to describe these changes in the network with numerical features. So they have this approach to classify these patterns of proteins in cells. So there are several features in terms of morphology. Euler number, size of the objects, variation of the size. Then we have edge-related features. There's shape-related, eccentricity, the convex hull, and the morphological skeleton. There are also these heraldic features, anchor moments. I haven't used those yet. And so what I did in this context was take sets of cells, let's say 30 cells for every condition, and look at the distribution of all these features before and after treatment. And then if we have a significant change, then we say that the feature is affected after treatment. And then we can do statistics on that, and for every feature see whether we have statistically significant difference or not. And then basically classify cells. So these are the cells, the blue is the nucleus, the microtubules are in green, and these are the cells before treatment. So they have, without treatment, they're different, which we can quantify as well. But also after treatment with the microtubule targeting drug, we see how there is changes in the organization. And so these are, for example, this is the most responsive cell, so we have changes in terms of 10 parameters. And then these three actually were resistant, and they change in 1 or 2 parameters. These three were sensitive, and they change in 4 or more.
What is blue in this?
DAPI, the nucleus. DAPI, the nucleus of the, yeah. So what we quantify here is only the green, the microtubules. But another thing to quantify in the next step is also the changes in the DAPI, in the blue. Because also the DNA gets fragmented from treatment.
So, I mean, given these images, then what? You want to segment them, and then you want to find some properties of each of this connected region?
What I want to have is like automated readout whether a cell is affected or not. And any presentation of the green area of the cell of this image will be a good readout. Like so far what I thought, for example, was that if we have certain edges with a certain angle, like let's say uniform angle of the edges, would mean that they're affected by treatment, because there is a reorganization of all the proteins. Instead of some spread proteins like that, they become all aligned in one. So in a way, I want to quantify how the image changes and how these individual microtubule fibers reorganize within the cell. And then if I have also, I'm pretty much at the end of that, but if we have like, in resistant cell lines, the images don't change. So they're treated, they don't change, so we know that we can say they're not affected. But also what is good about that, if we have a list of changes we can study, and in different cells that respond to treatment, different subsets affected, that would allow us to study different mechanism of drug action. So we can actually look for dissecting how did exactly the drug affected the cell.
So, but I mean, can you go back to the...
Yes.
So in this one, it's not necessary that you segment and identify the cell. You are looking at some properties like, orientation and angles and so on.
Let's say, yes. For example, this image. So here, this cell is not responding to treatment. So we have before treatment and after treatment, they look very similar. This one, for example, what we have is these fibers.
So you just want to say is responding, not responding, or that you can...
I want to put a number to it as well. Let's say we have, if you look at objects here, or if you do some thresholding, and we can identify here, let's say, these fibers and see how thick they are, how long they are, how many they are, what is the intensity of something compared to this, because this is like a spider net. Yeah. Very intricate and not very bright, and this is very different, right? And so we can say that they are affected to know, but also ideally we have a certain number to the level to which they're affected.
So can the doctor like give that number, like looking at?
No, they don't have a way. They just look. No. They can look at the images, but if you think about looking more in terms of a screening, like many cells, many patients, that becomes very difficult for a physician. And so that's the summary of this. This project is the idea is to work towards personalization of treatment by being able to predict the response to a perfect drug. So and then tailor therapy in real time. First, to have an enumeration readout to see if the cells are becoming less metastatic cells after treatment, and then to have a functional response. And this all work is done in Cornell Medical, mostly in the lab of my advisor, Evgen Ekaku, and also the 10x low resolution images were taken in the lab of David Nanus, and the high resolution in the lab of Manish Shah. So that's it. Thank you very much. I'll be happy to answer any questions.
Okay, any other questions? Yeah, go ahead.
The cell tracking work that you're doing, the videos that you have, what is the temporal resolution compared to the speed of the stuff that you're trying to track? Like the velocities of points in terms of frames.
So I don't remember off the top of my head exactly. So we have a little bit problem of the sampling because there is bleaching. So basically, the more often you image an event, the less you can observe it, sure. Right? Because the probe, they're fluorescent, so at some point they don't show light anymore. So the idea is, I think most of these images are taken every 5 seconds. So if you have very fast events, of course you want to take every 1 second, every 2 seconds in order to facilitate the tracking. But then you cannot observe them very long time, right? So the trade-off between.
Yeah, I asked because as the speeds of the points in terms of frames will increase. So if the speed of the object in successive frames is more than the distance between them, then obviously it'll be harder to track.
Yeah. That's why I had to use motion models. Yeah.
Yeah, go ahead.
So for the tracking, what are the features you use? Is it only the distance and the angle? Is there any other feature that you use for getting the cost?
I was thinking of using the intensity, but that really fluctuates because of the way the feature is formed, so that was not reliable. So I used the 2 angles for the local and the global, and the changes in the step in terms of.
Anything like the shape and other?
The readout is just XY coordinate because this is a sub-resolution feature. So basically, I identified as one pixel that is significant. It's a local maximum point that is significantly high above its immediate background. So there is no shape. It's more.
So what is the object size on an average? It's not 1 pixel, right? It should be.
It's defined as a sub-resolution feature, meaning that we have basically multiple fluorophores that are within the size of one single pixel, right? So it's really 1 pixel. Yes?
So that basically means most of your features are based on the motion and the spatial location, that you're not really using any appearance or anything to distinguish regardless of the spatial location motion. Not anything to distinguish between 2 cells based on, say, shape or any morphological based
shape on what?
On cells. Like when you're doing the tracking, so like angles and spatial XY, all these are like all based on the spatial location or say the motion of the cell, not say appearance. Whereas say like in say human tracking, like if.
But I mean that's what the answer. He's saying that since it's so small.
Yeah, that's
not much.
But like at some point in the like halfway through the presentation, you were extracting some morphological. It was for another problem for the cancer cells, but going to use something similar to that for the cells because even though they look very similar, but there seem to be like tiny differences in say size of them or say like how like even just the intensity, like even they're dark or bright as compared to the neighboring area.
So by the definition, it's a 1 pixel feature. So I see what you mean. problem is that there is really high density, right? There's really high density. If you look at, I'll show you just one movie, which I showed before, and if you look at that, and you can imagine how you would map right? So if you want to
For instance, in each of these pixels, like you see there are differences in intensity value of them, which is like the way like we as humans track them, or probably we take that into account.
So I think that the difference in the intensity is actually misleading, because you remember that these features are randomly the sub-resolution clusters of this fluorophore. So what you have here is a structure. So you think about this here. This is what the size of one pixel in the camera, and you have 440 dimers. So this is one dimer. So what you need to have is between 4 and 7 of them to be labeled to pop up. So if I have now 4 or 5, another one here is stochastically added, another one is removed. Also they moved. So every feature is lays on multiple fibers. So it's not only one. So which means that if they move left or right, any direction, you change the intensity. Yes?
Is the movement of each of these particles dependent on the neighboring particles? I mean, like for example, in crowd and humans, I mean, the way that you move is dependent on the, like, if it's really dense, it depends on the humans around you. I want to say the movement here is also dependent on the neighboring particles, or they completely move independently.
I mean, so this is one rigid structure. So if you have something here, and here they're going to stay constant throughout. But these two, even if they're next to each other, they can slide by molecular motors. So basically, there is some sort of coherence, but not completely, because this here is 25 nanometers. And the cutoff of the resolution is, let's say, 10 of them. So if there are 10 of them here on this length, and they can have different speed of motion as well, right? So
what I want to say, do you think that this contextual information can help you in tracking?
I thought no, because there's a lot of uncertainty. So you cannot have a model for...
So in this, I mean, when I watch this movie, from my point of view, I think the detection itself is a harder problem, because it's very difficult to, these points are so small, and the question is that, what you are detecting is simple approach of this difference of Gaussian and so on. All of those points are meaningful, or you just get these points and still overall it's an interesting thing to do. Like each of this point, which you are tracking, is it physically significant? Is that mean something?
So the meaning of this is not, so the way this all started is by synthetic simulation of that particle. So we can recalculate this system by a stochastic model, and then simulate this stochastic incorporation, because you don't know when a new label diamond will be added, when it's going to fall off. Mm-hmm. But the idea is that what the meaning of this individual speckles is, they show translocation of the polymer. So they show that there is motion, and the idea is that that's why I want to get, let's say, 10,000 or 100,000 of them, because in this distribution there will be enough informative particles. Otherwise, to go down to the individual one, I don't know how we can improve on the detection. It's always basically, and I have some movies when I've done actually movies when I subtracted the background so we can see only the bright ones. They still move in the same way. Mm-hmm. I think here for me the hardest problem in this is to dissect the antiparallel motion, because oftentimes we can see it by eye, but it's impossible to reliably track, because they are so close to each other and they move fast, and we cannot sample very, very, very often.
So Just as part of information, what's the point of doing the tracking? Is it to just get the flow? I mean, you know already there's an entire parallel flow. Is it to measure the velocity?
Yeah, so the flow is that it's the different mechanisms. So what you do in cell biology is you study how this system react, and also you have perturbations of the system where certain mechanism is impaired, something is accelerated. So what you want to do is to be able to quantify and to dissect and to understand what exactly happened behind. This one, for example. So what I saw here, for example, is that so this initially was thought of a zone where nothing happens. Just there is a network coming in. So this here is narrow. So you have a large site and a narrow. And so they thought here everything stops. And the idea is that if you see the yellow vectors that go up, they have faster distribution but high variation, which you can think it makes sense because you go from narrow to wider. Opposite these guys that go inside are slower and also very narrow. And so there is a lot of, there is disease, anything that you can study with such models because these are nerve cells and axons and neurodegenerative diseases. So especially in terms of treatment, right? You want to see how a drug affects the system dynamically in a living cell, not in a fixed dead cell.
So another question is, when you're doing this sort of imaging, in both the cell tracking as well as the tracking of the cancer cells in blood, for lack of a better word, is it like a moving sensor, or is the object, the actual physical object, moving with respect to the sensor or the microscope? I mean, the cells are obviously moving.
Yes. So you have a cell.
When the cell is on, is that moving?
No. You have a dish, and you put it under the microscope, and you still there, and inside the cell is just.
Right, but say you're studying cell division. You're looking at this entire flow and everything.
Yeah.
And that's within the cell. Is the cell itself moving or not?
They are moving, yes. So the cell division is not really a cell, it's an extract. So what they have done is they've broken the cell wall, so just to get the stuff outside so they can manipulate with these needles, anything like that. So they're not confined by anything. So they are moving around sometimes, yes. So that's the registration and alignment is additional problem, yes.
Oh, okay. Yeah,
but what we can do is you can image 100 of them and select the 10 or 20 that don't move as a first approximation and do some tracking. But some of them really move a lot, yeah.
Okay, yeah.
So in the case that you have like rotation or you have a source or sink of like the motion, how your algorithm can handle them? Because you do like you run your algorithm, then you do recursive runs to get the direction.
If they're moving, you need to do alignment, right, of the images. You need to align them and then detect and track. So you need to correct for the motion of the structure.
But like the sample that you had, the cells were kind of like rotating all around.
The base was fixed on the needle. So they're just, I was analyzing there is this lateral motion combined with the flux. I mean, you can dissect, the different components.
Okay, thank you.
Thank you.